Hello and welcome to the Human Cell Biology unit. So this is the 2024 route through for all of the Faculty of Science. And this is a new unit. So as always, please check the unit code to make sure that this is applicable and you have the right assessment form. So in this session, we'll be covering looking at the structure of eukaryotic and prokaryotic cells, understanding the function of cell organelles, and understanding how materials are exchanged across the cell membrane. During our session, there's a couple of key little graphics which I'm going to introduce. So they are fairly self-explanatory. You can pause the screen now and just take a quick look at those, and I'll explain each of them as I go through. So the first learning outcome for this unit is understanding the structure of eukaryotic and prokaryotic cells. So in the next set of slides, I'm going to go over in detail what a prokaryotic and eukaryotic cell is, and then you may wish to use the thinking maps to help you map out the key differences between those two types of cells. So, cells are fundamental building blocks of all living organisms, from unicellular cells of bacteria to much more complex cells that make up human beings. Every living thing is made up of cells. And these microscopic units are not only the basic structural units, imagine like the basic building blocks of life, but they're also functional units. They carry out essential processes which sustain life. And we can categorize them into two categories: prokaryotic cells and eukaryotic cells. I'm going to start by explaining prokaryotic cells because these are the most ancient forms of life, dating back over 3.5 billion years ago. They're simple cells. They have no nuclear. They have no nucleus present. The genetic material is just located within the nucleoid. And that's what the name actually means. Pro meaning before, and karyote meaning nucleus, before nucleus. So the genetic material is not bound within a nucleus. And examples would be, for example, bacteria and archaea. And these are much more simple cells. And we'll come back to that later on comparing them. Eukaryotic cells, and if you take the prefix of that word, eu meaning within a nucleus. So these are much more complex cells. They include animal cells, plant cells, fungi, protists, and they all have nucleus which houses that genetic material. And they've also got various other membrane-bound organelles. Our organelles are all of the little pieces within the cells. And I'm going to direct you to a great little video that I use with GCSE students. I love it. And it's the cells wrap. And they literally go cells, cells. They're made of organelles. And it's because the organelles are all of the individual little pieces of the cell which work together for it to function as a cell. So our eukaryotic cells are much more complex. And so if we liken them to an office space, imagine prokaryotic cells would be a little bit like an open plan office with a load of furniture just chucked in, some desks, some computers, some chairs, kind of not very well organized, just bundled in there, open plan space. Whereas a eukaryotic cell are much more complex. That'll be much more like a building of office spaces, which are dedicated to perform certain roles, specialized in each room, performing a different task, and areas associated with eukaryotic cells are linked to different processes. So why do we need to know this? Well, actually, it's really fundamental. If you understand the cells which act within different groups of tissues, then that can obviously have huge, really, really important implications when studying like life sciences, but also really important to help us understand how we evolved. So life evolved from these very simple cells into these much more complex forms. And by studying all of these cells, we can have a much greater insight into that. So for this activity, I want you to pause the video and stop and reflect what you have learned about the difference between prokaryotic and eukaryotic cells. And I'd like you to use this tool called a double bubble map. And this is one of the thinking maps which I've uploaded into the Faculty of Science area. The reason I use these maps is because they're based on academic research that came out of Exeter University when they looked at the most effective ways of people processing information when they're studying to maximize their learning. So these are evidence-based resources, and you'll probably recognize it's a little bit similar to a Venn diagram. So when you do a Venn diagram, you have the two circles that overlap. The individual circles are the things that are unique to the two things you're comparing, and then the overlap obviously is the similarities. I think the beauty of using these ones is it makes you a little bit more concise, because you're going to map out the key things within your research and your work, so that you're not just like writing down everything. You need to be quite concise. And then what that will do is it will allow you to have the key ideas, and then you'll be able to compare and contrast the two types of cells in extended writing form much more simply. So you can see they do work a little bit similar to if you've used a Venn diagram before. So you're going to literally write in, I've just put the pictures in there because I thought it'd just be easier to fit them in there. So prokaryotic. And if you're printing these off, obviously they're available as PDF images, they're available as JPEGs, all in the folder. But you can either write in the name, or if you wanted to, you could stick on a picture. If you're doing that electronically, that'd be quite easy to cut and paste. And then you're going to put the key characteristics. So obviously the circles that only have one linking line are the characteristics that are unique. So the far left boxes are the characteristics that are unique to prokaryotic cells. So just to start you off, these are the most simple and ancient types of cell, extending to those that existed over 3.5 billion years ago. So that would be a characteristic which is unique to prokaryotic cells. Then you'd have a look at similarities. So obviously the three arching boxes in between the two bubbles that you're comparing, obviously they're features that both of the types of cells will have. And then obviously on the very far right, there's another three bubbles that are solely attached to eukaryotic cells. And again, we'll be looking for features that are unique to cells which are categorized as eukaryotic cells. So if you want to pause the video at this point, reflect on the things that I've gone through in the previous slides, and you may want to do some additional research to add to your notes, to add a bit more depth. Places that I'd suggest that you go to do that research would be something like Britannica. I'm going to pop a link on the reading list for Britannica for prokaryotic and eukaryotic cells. The reason I'm suggesting Britannica as a good academic source of information is that it's constantly reviewed by an editorial team. It's easy for referencing. You can literally click on the citation. You can see exactly who's edited it and when. And also it's got a really nice level of depth to the explanations, but it's also really accessible. So enjoy that task. Here's a table of some suggested features, which you may have found either from the previous video clip or from further research. You can pause here and just collect and add any that you maybe didn't get, or maybe some that you want to explore further for your extended writing answer that you'll add into your report. So in the assignment brief, one of the tasks is that you are able to identify this specific set of cell organelles. So remembering an organelle is a small component which is part of the cell which performs a function which contributes towards the cell being living and operating. So you need to be able to identify the nucleus, the cell membrane, the endoplasmic reticulum, the ribosomes, the mitochondria, the Golgi body, and the lysosomes. Now, you need to be able to do this on a micrograph. And all that is an image or photograph that's captured using a microscope. So it allows us to view images and structures which would be far too small to be seen with the naked eye. Now, just to put this into context, you probably used a light microscope before. So a light microscope would probably allow us, for a plant cell, to be able to see potentially the nucleus with a dye. But an electron microscope would allow us to see much more intricate structures. So there's various different types of microscope, and the main key difference is that they are able to see different magnification and different components and parts of the cell. So here is a little image as an example, and it's been labelled with the organelles on there. I want you to replicate this. I want you to find your own micrograph image. You're going to use a diagram as a point of reference, and then you're going to use it to be able to identify the different organelles within that eukaryotic cell, and that is the task. So one of the tasks on the assignment brief is that you're able to calculate cell or organelle size using this calculation. Now, the reason I've put it into this little triangle is it's a really simple way for you to be able to rearrange the equation so that you can make the subject that you're trying to find out the subject within the equation. So if you put your finger over what you want to calculate, so for example, image size would be the actual size times by the magnification. The actual size, meaning how much it actually physically measures in length, is the image size, so whatever you're measuring it on the page as, divided by the magnification. So you need to take an image which obviously hasn't been altered in size. I've popped a little ruler to scale on here. And so I'm going to take it from here. So I'm measuring the length of the mitochondria. I've placed the ruler on the beginning of the mitochondria, which is the little yellow and orange stripy organelle within the cell, and I measure it at 15 millimeters. Now, of course, we need to convert this to micrometers. And so there's 1,000 micrometers within each millimeter. So in which case, 50 millimeters would equate to 15,000 micrometers. And it's depicted using that little symbol, which looks like a U with a long part on it. And so to work out the actual size, we're going to do our image size divided by magnification. So in this case, it's 15,000 micrometers divided by 30,000. And so the actual size of the mitochondria is 0.5 micrometers, which actually is within the limit of what you'd expect it to be. So what I've done is you can either find an image with, obviously you're going to need two pieces of data, okay, to be able to calculate the actual size of it. And you're going to talk through in your report the image, the calculation, and where you got the numbers from, and do it as a staged calculation explaining each stage. Now, if you're struggling to find images with the right amount of information on them, then I have downloaded a whole load of exam questions, which you can pull the information from, and that will have realistic data which you can use in your reports. Okay, so if there's any problems with that, let us know. I can do an individual tutorial on this specific math skill if this is something that you find challenging. In this next section, we'll be looking at the functions of the different cell organelles within eukaryotic cells. So we're going to focus on the nucleus first, the first organelle which is contained within the cell. And so the nucleus is made of three individual parts. I'm going to start with the nuclear membrane. So the nuclear membrane is a double-layered membrane which surrounds the nucleus, and it protects all of the DNA which is contained within the nucleus. So the nucleus contains chromosomes. Those chromosomes are wound tight storage mechanism of DNA, long strands of DNA. And in a classic human cell, we'd have 23 pairs of chromosomes, one from mum, one from dad, so 46 in total, with the exception, obviously, of sperm and egg cells, which only have 23. So this nuclear membrane surrounds the nucleus, keeping all of that DNA inside safe. The DNA does direct the cellular activities, but also contains a genetic map, which basically is an instruction guide to make a full version of you. And as you probably know, your genes are unique to you. So then we have our nuclear pores. Our nuclear pores are scattered around the nuclear membrane, and there's numerous small openings. Okay, now these small openings are crucial to controlling what moves in and out of the nucleus. And they allow the passage of important molecules like RNA and ribosomal subunits, which are needed for a process called protein synthesis. If you think synthesis means to make and protein, so literally the making of proteins, DNA is a protein. And so the constant process of protein synthesis means that, to make more DNA, which cells need to do before they replicate, this process needs those molecules moving in and out of the nucleus and making sure that the DNA is secured within the nucleus. We then have another area of the nucleus called the nucleolus. And this is within the nucleus, and it's a dense region. That dense region is where ribosomes are assembled. And those ribosomes are essential parts of the protein synthesis process, and they're responsible for translating genetic information from RNA into proteins, and they carry out various functions throughout the cell. So, in summary, the nuclear membrane protects genetic information. The nuclear pores regulate the exchange of materials, and the nucleolus is where the ribosomes are produced. And together, all of these structures work effectively to control the cell's activities whilst safeguarding the genetic blueprints of the cell. So we're going to hit 3 different organelles within this video. So we're going to look at the 2 different types of endoplasmic reticulum, rough ER and smooth ER, and ribosomes, because those little rough bits on the rough ER are ribosomes. So the endoplasmic reticulum is a crucial organelle. It comes in 2 forms. So the rough, the purple one with the little dots on, which are just ribosomes, and the smooth, which is the pink version. Obviously, there's no actual colour difference. I just want to differentiate between them. And then we're going to have a look at the ribosomes. So the rough ER is covered with ribosomes, and that's what gives it that bumpy appearance, hence the name. And it can be viewed under a microscope. The ribosomes are the cell's protein factories. So they synthesize the proteins, and the rough ER provides a site for these proteins to be folded and processed. And once the proteins are properly assembled, the rough ER packages them into little vessels and it sends them to the Golgi apparatus. And then that's directly sent to their final destination within the cell. In contrast, the smooth endoplasmic reticulum contains no ribosomes. So it lacks any ribosomes, which gives it that smooth appearance, but it actually plays a different role. It focuses on synthesis of lipids linked to fats and detoxification of harmful substances and storage of calcium ions. And so obviously they're also essential processes linked to cell processes. So then finally, the ribosomes. So the ribosomes are these very small granular structures found either attached to the rough endoplasmic reticulum or floating freely in the cytoplasm. And they're made up of 2 subunits that are responsible for translating genetic information from messenger RNA, which is literally called mRNA, and into a specific sequence of amino acids to build proteins. And then these proteins go on to perform functions within the cell, such as acting as enzymes, structural components, signaling molecules. They're all proteins. So in summary, the rough ER works closely with the ribosomes to synthesize and process proteins, whereas the smooth ER handles lipid production and other vital tasks. But together, these organelles ensure that the cell can produce the proteins that it needs and allow all of those processes to allow the cell to function properly. So the next organelle we're going to look at is the mitochondria. So the mitochondria are the site of aerobic respiration. And the process of respiration is the process which generates energy that the cell needs to perform all kinds of functions. So we'll focus firstly on the structure of the mitochondria. So mitochondria have this unique structure, which is key to their role in energy production. They have a double membrane, and the outer membrane is smooth and acts as this protective boundary, while the inner membrane is highly folded, and that's why it looks like a little comb structure. I always think it looks like a little comb anyway. And so it's highly folded into structures known as cristae. And these folds increase the surface area, allowing for more space for the process that produces energy. And then the inner membrane is like a matrix, a gel like substance that contains enzymes and mitochondrial DNA and ribosomes. So that's what the structure looks like. So its role. So the role in aerobic respiration, and what we mean by aerobic respiration is in the presence of oxygen. So in order to produce energy from respiration, you need oxygen and glucose, and they are then converted, and ATP energy is released. And as a byproduct, the waste products are carbon dioxide and water. So, the primary function of mitochondria is to convert energy stored in food molecules into that molecule that I mentioned, ATP, which is adenosine triphosphate. And so that's the main energy currency of the cell. And this process is known as aerobic respiration, and it occurs in several stages, including glycolysis, the citric acid cycle, and the electron transport chain. And these cristae in the inner membrane are where the electron transport chain takes place. It drives that production of ATP. So essentially, the structure of mitochondria is specially adapted to perform its role in energy production. The inner membrane provides that big surface area for ATP synthesis. And then the matrix hosts that critical conditions for the reactions of the citric acid cycle. And then without these mitochondria, cells just wouldn't be able to produce enough energy needed to perform all the functions of the cell. And then obviously, all of those cells producing energy in tissue, then obviously provide that energy for organs to work, heart to beat, etc. So the next organelles that we're going to look at are the Golgi body and the lysosomes, and they're two essential organelles that play a crucial role in the cell's internal logistics and maintenance. So we'll start off with the Golgi body, and it's also known as the Golgi apparatus or the Golgi complex, and it's a series of flattened membrane-bound sacs called cisternae, and it acts as the cell's post office, where proteins and lipids are synthesized by the endoplasmic reticulum and then further modified and sorted and packaged. And once those proteins reach the Golgi body, they may undergo various modifications, such as the additions of sugar molecules in a process called glycosylation. And afterwards, then the Golgi body packages those proteins into vesicles, and then they're transported to their specific destinations outside of the cell. And this precise packaging and modification process is vital for making sure that those proteins function correctly and reach the right location. So lysosomes, on the other hand, so they're the cell's waste disposal and recycling centres. So these little small membrane-bound vesicles contain powerful digestive enzymes, and that just digestive enzymes break things down into smaller molecules. So they're capable of breaking down various biological molecules, including proteins, lipids, and carbohydrates and nucleic acids. And so lysosomes are responsible for basically digesting, and imagine like you've got your big boxes and you're packaging, ripping them down into smaller parts, a little bit like that. So the lysosomes are responsible for digesting and recycling those cellular debris, those damaged organelles, those even pathogens like bacteria that invade the cell. They break them down into materials, and lysosomes help maintain the cell's health and prevent the buildup of waste products that would otherwise be harmful. So, in summary, the Golgi body ensures that proteins are correctly modified and delivered to destinations, whereas the lysosomes keep the cell clean by digesting any unwanted materials. But basically, the two work together to maintain the cell's functionality and integrity. So now we're moving on to the cell membrane, which is also known as the plasma membrane. So its dynamic and flexible barrier surrounds the cell, providing both protection but also controlling what goes in and out of the cell, which is really important, obviously, for those processes. So the phospholipid bilayer, is the bit that we're going to focus on first out of the four structures that we're going to look at. And the phospholipid bilayer is composed of these molecules which have a hydrophilic water attracting head and two hydrophobic water repelling tails. In the bilayer, the hydrophobic tails face inwards, shielding from the water, whereas the hydrophilic heads face outwards, interacting with the aqueous environment inside and outside of the cell. So this arrangement forms this semi-permeable, meaning that some molecules are allowed to pass through and others are not. So it creates a semi-permeable membrane, only allowing certain molecules to pass. So the next part of our focus on the cell membrane, we're going to have a look at cholesterol. So cholesterol is interspersed within that phospholipid bilayer. And so those cholesterol molecules, which are basically just spread out through, play a critical role in maintaining the fluidity of the membrane. So making sure that it remains flexible but stable at different temperatures. And it also helps to prevent the cell membrane from becoming too rigid or too permeable. Permeable means like how easily things can pass through it. So then we've got our third focus, which is the proteins. So embedded within that phospho bilayer are various proteins which serve multiple functions. So some of the proteins are known as integral proteins, and they span the whole membrane and form a channel or a pore that allows specific molecules to move in and out of the cell. Otherwise known as peripheral proteins, they're attached to the surface and they play roles in signaling and maintaining the cell shape and anchoring the cell in place. So you can see in the little diagram, there's some different examples of those proteins. And then we've got our glycoproteins and our glycolipids. So on that outer surface of the cell membrane, you'll find glycoproteins and glycolipids. These molecules are where carbohydrates, sugars, are attached to the proteins and lipids, respectively. And those glycoproteins and glycolipids are crucial because the cell needs to be able to recognize and communicate. And so they act as like little identification, little markers, which allow cells to recognize each other, which is really important for immune response and tissue formation. So, in summary, the cell membrane structure has that phospholipid bilayer, really important, those hydrophobic and hydrophilic areas to allow that aqueous movement of solution. They've got the cholesterol, which obviously allows it spread out and really important in maintaining that structure and allowing that fluctuation in temperature. And then you've got the proteins, which create those channels, but also create those specific areas to signal. And then we've got our glycoproteins and lipids, which act as markers for cell signaling and immune response. For the final third section of the assignment, we're going to focus on how materials exchange across cell membranes, looking at the processes that are involved in the exchange of materials across that cell membrane and the structure of it. Okay, so as I mentioned in the previous part about the cell membrane is not just a barrier, it regulates the movement of those substances in and out of the cell. So we're going to have a look at some different types of processes which move things in and out of the cell. So, first is diffusion, and this happens when you light a candle or spray perfume in a room. It goes from an area that it's very concentrated, so where you've just sprayed the perfume, there's the most perfume molecules, and naturally it will move from an area of high concentration into an area of low concentration because the molecules, the particles are constantly colliding and moving in the air and moving outwards. So it'll move from an area of high concentration into an area of low concentration. And hence, like if someone sprays some perfume on a train, you might smell it down, part way down the carriage because the smell has diffused, but it won't be as concentrated as where they first sprayed it. The next type of movement of substances is osmosis. Osmosis specifically refers to the movement of water. The movement of water molecules from an area of high concentration into an area of low concentration across a semi-permeable membrane. So the process of osmosis only refers to water, the movement of water. We've then got the movement of molecules by active transport, and this actually works in the opposite direction. So unlike diffusion and osmosis, active transport requires energy. So it needs energy, and because it is moving against the concentration gradient, so it's moving from an area of low concentration into an area of high concentration. And this process is crucial for maintaining concentrations of iron, such as sodium and potassium, in and out of the cell. The final type of movement of substances is about large molecules being put in or out of the cell using those vesicles, which we talked about earlier. So cytosis is a process that involves the movement of large molecules or particles in or out of the cell by packaging them in these little vesicles. There's two main types. So there's endocytosis. So this is where a cell engulfs a material from the external environment and encloses them in a vesicle, which pinches them off from the membrane and brings the material into the cell. Or exocytosis. So this is the reverse of endocytosis. Exocytosis involves the vesicles fusing with the cell membrane to release their contents outside of the cell. And this is how cells excrete substances like hormones and neurotransmitters to get rid of them. So that was kind of moving things in and out of the cell, but packaging up bigger things. So together, all of those mechanisms, so diffusion, osmosis, active transport, and cytosis, all make sure that the cell can efficiently exchange materials with the environment. And this is to maintain a process called homeostasis. So you probably are familiar that the body needs to be kept at a temperature not fluctuating much from 37. There's specific conditions within the body which need to be maintained. For example, your glucose levels. So obviously, if your glucose levels plummeted, then you start to feel dizzy, faint. Okay, so homeostasis is all about maintaining those conditions within the body, and the cells play a crucial role in that. And they can respond using these mechanisms to changes in the environment. So I'm just going to do a very quick recap of the structure of the cell membrane and how it helps that material exchange, because I covered that fairly well detailed in the description of what the cell membrane is. So remember, we've got those phospholipid bilayers. So the semi-permeable nature of that phospholipid bilayer, it makes it really, really simple for diffusion. So it allows the small non-polar molecules to pass through really, really easily, while larger polar molecules are restricted. It's got those integral proteins. The proteins are crucial for facilitating diffusion and active transport. And then those channels and carrier proteins embedded in the membrane, which allow selective passage of ions and molecules. And then in our active transport, these proteins also use energy to move substances across the concentration gradient, against the concentration gradient, Then we've got our aquaporins, and these are specialized channel proteins which facilitate osmosis, allowing water to move rapidly into the cell membrane while maintaining the balance of fluids inside and outside of the cell. Because remember, obviously, if cells all of a sudden have too much water that moves into them, they would swell and burst. And then we've got cholesterol. Remember, that was really key in making sure that the bilayer maintains that fluidity, and it's also essential for processing processes like endocytosis and exocytosis. Then we've got finally those glycoproteins and glycolipids, and these molecules play a role in the cell recognition and signaling, which influence the transport process and actually the triggering of that endocytosis, which specific molecules bind to the receptor on the cell surface. So, in summary, that cell membrane structure is composed of phospholipid bilayers, protein and cholesterol, and carbohydrate chains. And basically, it's perfectly adapted to facilitate and regulate the movement of substances in and out of the cell for all of the processes that are occurring in the cell. Okay, so this is the final little bit from me. So really, really important that you go through your study plan, check your assignment module title and the unit code, and make sure that you've definitely got the correct assignment brief in front of you. All assignment briefs will have the learner outcomes, which we've gone through today with the content, and then the assessment criteria. Use this as a checklist for yourself. Make sure that you have demonstrated each and every one of those skills and pieces of knowledge that they expect. Now, obviously, you're going to use these alongside the graded criteria. So, the graded criteria are further down in the assignment brief. So, if you were to meet each of these points 1.1, 1.2, 1.3, etc., then that will be a pass. But then, if you demonstrate a higher level according to those graded criteria, then you can achieve a merit or a distinction. Okay, and you need to look at your course really carefully to see how many, for your university course, how many merits and distinctions they need from the course overall. And it's really good to know that early on. So when you're going through each of these criteria, so let's go through the first one, 1.1, describe the structure of eukaryotic cells and identify the nucleus, the cell membrane, the endoplasmic reticulum, ribosomes, mitochondria, Golgi body, and lysosomes from electron micrographs. An electron micrograph is specifically an image which has been taken using a microscope. And so, a little bit like the image that I modeled for you, and it had all of the different areas labeled. So, you're going to want to find yourself a nice diagram of a eukaryotic cell and where you'd find those individual organelles. And then you're going to use that to help identify on the image that you find. The next point, compare and contrast prokaryotic and eukaryotic cells. Well, earlier in the video, I suggested that you completed a bubble thinking map, a bubble map, double bubble map, where you're comparing prokaryotic and eukaryotic cells, and that will support you in your extended writing. For this assignment, you're going to produce a report. So I would do these as have little side headings. The way that I'd probably structure it, I'd probably have this report is going to investigate and look at the importance of different structures related to their functions in cells. So I'd maybe have a side heading for the first one, structure of eukaryotic cells. I'd have my image, I'd have all my annotation of my names labeled, going through and ticking off. I think I made a table in there so you could tick off as you go through. Then I'd go on and say there are two main types of cells, prokaryotic cells and eukaryotic cells. I'd explain a little bit about that. Then I'd go into my discussion about what the key differences are between prokaryotic and eukaryotic cells. And then I'd be looking at, okay, how do I measure them? So you're going to find your image. There's some examples for you to work through. You maybe want to take one of those and you could then show your working. But you have to demonstrate that you're able to estimate the size of a cell or an organelle from those images or a study. So you're going to go through each of these criteria and make sure that you've met them. I would start by making your notes from the video. And then I would go and research further the bits that perhaps you need to flesh out a little bit, the bits that maybe you don't remember so well from GCSE. Maybe you want to spend a little bit of time reading extra on Britannica. They've got some great pages of information, which are easy to cite as well. And then the assignment itself can be no more than 2,000 words. It is a 3 credit graded units, and therefore the total word count can be no more than 2,000. And if it is, and then it's externally moderated and it's over the word count, then it'll be capped at a pass. So that's really, really important. You don't go over the word count. And it's a really important skill to be able to make sure that you're concise in your explanations. And 2,000 words, to be honest, is more than enough to cover all of these sections. If you've got any questions, drop me a message in the chat, and we'll have some seminars to discuss this further. Good luck with your assignment, and I can't wait to read them.